Good morning. We'll start promptly in 1 minute at 11 a.m. We'll begin shortly. Okay, it's 11 a.m. We'll prepare to begin the program.
Good morning. I'm Gail Jarvik, 2021 President of the American Society of Human Genetics. Welcome to the presentation of the 21st Annual Gruber Prize, awarded for groundbreaking contributions to any realm of genetics research. We are pleased to host the Gruber Foundation and invite their Executive Director, Sarah Rea, to say a few words about the foundation and the prize. Sarah?
Thank you, Professor Jarvik. Let me just share my screen and start my PowerPoint. Great. We are delighted to participate virtually in the annual meeting of the American Society of Human Genetics to honor Stuart Orkin, who joins both an illustrious genetics laureate list and also the recipients of the 2021 Cosmology and Neuroscience Prize on our annual roster. If you are considering nominating for a Gruber Prize, note that we encourage nominations that reflect the breadth of the fields and the diversity of those working within them. The submission deadline is December 15th. Before we return to genetics, I must acknowledge our co-founders, Peter and Patricia Gruber, whose combined vision and leadership established the International Prize Program, and whose care in doing so gave it the legs to stand on its own. Dr. Orkin was chosen by a distinguished selection advisory board nominated by the Genetics Society of America. Let me invite the GSA president, Hugo Ballin, to say a few words.
Good morning. I'm Hugo Ballin, president of the Genetics Society of America. GSA partners with Gruber on the Genetics Prize, recommending the expert advisors that maintain scientific integrity. We're also pleased to partner on the Rosalind Franklin Young Investigator Award, created to honor the groundbreaking contribution of Dr. Franklin and to inspire and support new generations of women in the field of genetics. The 2022 Rosalind Franklin Fellows are Odie Bernheim, Kara McKinley, and Vivian Sloan. Thank you.
Thank you, Dr. Bellin, and congratulations to the Rosalind Franklin Fellows. The Gruber Foundation appreciates its relationship with GSA, just as we sincerely appreciate the knowledge, commitment, and enthusiasm that the selection advisory board members bring to the judging process. Let me now invite the chair of this board, Alan Spradling, to introduce the scientific accomplishments of our recipient and read the official citation to the prize. Professor Spradling?
Hi, I'm Alan Spradling. All of us engaged in basic biomedical research are driven by the prospect of discovering and elucidating new aspects of the universe and of humanity. Stuart Orkin has done this many times through his brilliant studies of the blood system that sustains our lives from the moment blood cells first form in the fetus to our final heartbeat. But most basic scientists scarcely dare to dream their hard-won findings within their lifetimes will make a difference to the well-being of patients, or that they might have a direct hand in bringing fruitful applications of their ideas to a patient's bedside. Stuart Orkin is on the threshold of this achievement as well, and we look forward to having the chance to hear him describe some of this work. Here's the award citation. The Gruber Foundation proudly presents the 2021 Genetics Prize to Stuart Orkin for his pioneering discoveries of the genetic and molecular basis of common blood diseases and the gene regulatory mechanisms that govern blood cell development. He elucidated the mechanism of regulation of the switch from fetal to adult hemoglobin and reduced to practice the reactivation of fetal hemoglobin as a therapeutic strategy for sickle cell disease and the thalassemias. Stuart Orkin's science has beautifully illustrated the successful progression from fundamental genetics to mechanistic biology to therapeutic development and intervention, which can transform the lives of millions of patients in need. Stuart Orkin, please come forward to receive the prize.
Professor Orkin, would you just push your prize pin forward a little?
Okay, I just want to show the pin, which is right here. It's a little hard to do in the screen.
That's what we would have put on you in person if we were together.
And this is the paperweight medal. Two sides, one says the Gruber Prize on it here.
And before you begin, let me just tell our audience that they may submit questions for the discussion via the Q&A function at any time during the lecture. Now I will turn things over to our laureate.
Okay, thank you. Before I begin with any slides, I want to thank the Gruber Foundation for this wonderful recognition of our work. And I mean our work, as the work being recognized is a joint effort of many trainees, colleagues, and collaborators. No investigator does it alone. I'm truly humbled as I look over the roster of extraordinary prior recipients of the award. I'm honored to join their ranks. This award also recognizes a field, that of the genetics of hemoglobin, which is quite old in the sense that has been the focus of investigators for many years, long before the era of cloning, PCR, and CRISPR. Extraordinary advances have been made in our understanding of these disorders under the spotlight today. Yet the overall burden of disease here in this country and worldwide remains to be reduced. Because one of the disorders, sickle cell disease, disproportionately affects those with more limited resources and those who live in resource-limited countries, parts of the world, investment in strategies to reduce this disease burden have sorely lagged. The recognition by the Gruber Foundation of the research I will review today is critical to bringing attention to the field. As I think was Alan Spradling said in his introductory remarks, we all have a common goal, and that is to use the tools of modern genetics, not only to understand biologic phenomena and disease in molecular terms, but to formulate mechanism-based therapies aimed at durable and transformative clinical outcomes. In this regard, we've been studying the hemoglobinopathies, of which there are two major types: beta thalassemia, which is a reduction of beta globin production and hemoglobin synthesis, and sickle cell anemia, a point mutation in the beta globin gene. Sickle cell anemia is illustrious in genetics. It's described more than 100 years ago, but Linus Pauling coined it the first molecular disease in 1949. And then Vernon Ingram, 7 years later, identified the specific amino acid substitution. Hemoglobin has really been a center of research in genetics throughout really the evolution of genetics. And this was really brought home by the comments of Arno Motulsky, one of the great geneticists, where he said hemoglobin research plays a role in human biochemical genetics similar to that of Drosophila research in formal genetics. And many fundamental concepts have become clarified by investigations of human hemoglobins. I just want to remind everyone that although we know quite a bit about these disorders, particularly sickle cell disease and thalassemia, they're really still a global health burden. As emphasized by the late David Weatherall, they're a global health burden that is not only present, but will increase over the coming years. And the World Health Organization estimates there are at least 3,000 new births of sickle cell disease children, largely in the African continent. There are about 75 to 100,000 individuals with sickle cell disease in the US. The crux of what I'll be talking about today is really illustrated here in the switch from fetal to adult hemoglobin. On chromosome 11, as shown at the top, the genes encoding one of the subunits of hemoglobin, the beta-like subunit, are encoded. And the locus, which has been studied since the first days of molecular biology and cloning, encodes an embryonic gene, 2 fetal genes, and the adult beta globin gene over approximately 50 kilobases of DNA. These genes are controlled by the locus control region, a super enhancer shown on the left. During development, there's a switch from gamma to beta expression. This is the fetal switch, which begins to occur in the 3rd trimester, but is completed by about 6 months after birth, as shown in this figure. And in adults, we have about 1% residual fetal hemoglobin expression, so it's not completely extinguished. This is the time course of hemoglobin F expression over development from 6 weeks, 12 weeks, 6 months after birth to show this progressive decrease. And it was Janet Watson, a pediatrician in 1948, who suggested that hemoglobin F was protective in sickle cell disease because young infants destined to have the disease phenotype as they grew older were not affected in the neonatal period. And she suggested that hemoglobin F was protective, which is correct. And a natural history study carried out many years later with support of the NIH showed that survival in sickle cell disease is greater with increased fetal hemoglobin. And this would be the same in beta thalassemia as well. So the therapeutic vision in this field for several decades now has been that if we could increase that 1% level of fetal hemoglobin in adults to perhaps something on the order of 20% evenly distributed among cells would be therapeutic, and that is would reduce sickle cell crises in sickle cell patients and reduce the need for transfusions in those with beta thalassemia. This is an old field in a sense that it's been under the microscope for many years. And I like this quote from Marcel Proust saying the only real voyage of discovery consists not in seeking new landscapes, but in having new eyes. And the evolution of technology and genetics provided these new eyes over time. This is a timeline of research on fetal hemoglobin, roughly from 1948, from the time of Janet Watson, through to the early 2000s, just after the turn of the century. I just want to emphasize that there are many important discoveries during this time, including the sickle cell mutation, discovery of transcription factors regulating genes in erythroid cells, the identification of the locus control region and chromosomal looping to regulate the globin genes, and the discovery of hydroxyurea as at least a partial therapy in sickle cell disease. But in this period of time, we also were sort of wandering in the desert, unable to really explain how this switch occurred. And really the crux of this is in a sense quite simple in molecular terms. It's how does the LCR, which engages the gamma genes during fetal liver development, how does it switch to link up with the beta globin gene, the adult gene in adult bone marrows, adult cells, and how are the gamma genes, the fetal genes, shut off in their expression? And this is a challenge that we've addressed over the last number of years. And really the breakthrough in this area was the introduction and application of genome-wide association studies, which are well known to this audience, but was depicted here, where population hemoglobin F is measured in a population, and SNPs across the genome are employed to try to find correlating loci. And there are really two of these studies that were important. One out of Swilathene at the end of 2007, which was really a rather small study suggesting there was a new gene on the chromosome 2 on the short arm, which was correlated, SNPs were correlated with fetal hemoglobin level, in this case actually F cells, the number of cells that express F in the blood. And then a larger study, which was that of David Schlesinger and the late Antonio Cao in Sardinia, with whom we collaborated, which was a full GWAS in that population. These studies had the same outcome, and that is that there are 3 loci associated with fetal hemoglobin levels: the beta globin cluster on short arm chromosome 11, which was, I think, already known, HBS1L MIB on long arm of chromosome 6, which is essentially the MIB oncogene, which controls really differentiation of cells, not control of fetal hemoglobin per se directly. And the new player was on the short arm of chromosome 2, BCL11A, which is the gene we focused on. And if one sums the genetic contribution of these 3 loci, it accounts for approximately 50% of the genetic variation in hemoglobin F, which is quite huge in these GWAS studies. The challenge in GWAS is obviously going from the entire genome of 3 billion base pairs to a few base pairs, which tell us something about the gene or the disease itself. The first step in this, which was performed by Vijay Sankaran, who was an MD PhD student at the beginning of these studies, was to examine the potential of BCL11A as a candidate repressor. And the first thing we noticed was that BCL11A is a stage-specific factor. That is, it's expressed in adult cells, that is in fetal liver and in bone marrow, but not in the primitive cells of the yolk sac, which are embryonic. And this suggested the factor was in fact a stage-specific transcription factor. Using a system of in vitro differentiation of primary CD34 cells, which allows one to look at erythroid maturation in culture, Vijay showed that use of an shRNA to inhibit BCL11A RNA boosted the level of fetal hemoglobin, and we suggested at this time that BCL11A might emerge as a new therapeutic target. This was a rather far-reaching conclusion at that time based on this evidence, but I think it's been borne out over the years. The critical question for us initially was how important is BCL11A as a single factor? Might there be many factors that would control fetal hemoglobin level, and therefore it'd be hard to reduce to practice perhaps a single target? Or was there 1 or 2 factors that are important? And to get at this, we used genetically engineered mice, of which there are 2 versions, so-called Berkeley mouse and the Towns mice, which are essentially interchangeable. And these mice have a knockout of the endogenous mouse globin genes, and human globin genes, including the sickle gene, have been introduced to recreate sickle cell disease in these mice. And these mice have a phenotype close to that of sickle cell disease. And we performed a rather simple experiment, which was to ask if we knock out BCL11A in the erythroid lineage alone, in this background of the sickle mouse, what would be the phenotype? Would we rescue this phenotype? And we restricted expression to the erythroid lineage because we knew at the time that BCL11A was expressed in other cell types, and we didn't want to confound the analysis. The rescue experiment was quite dramatic, more dramatic than we might have imagined. The wild type smear, blood smear shown on the left, in the middle is the sickle cell mouse with the really pathognomonic sickle forms and young red cells, which are these purple cells. But the intercross mice had a normal blood smear, essentially normal hematology. And the important finding was that they had a large activation of fetal hemoglobin expression, roughly to 25 or 30%, which is enormous compared to the sickle mouse background. And this fetal hemoglobin was evenly distributed in cells largely, in other words, pancellular, as one might anticipate from this genetic experiment. So this suggested that alone, BCL11A could be a target for rescue or reversal of the sickle cell disease phenotype. And this is really the basis for all the translational studies that have followed. Now, in the interim, we've learned there's another repressor of fetal hemoglobin called LRF or ZBTB7A, which is about equally potent, for which there's some genetic evidence, but BCL11A, we think, is probably the major physiologic regulator. I don't have time to go into the specific reasons for that today. BCL11A is a large protein, not easy to work with, which has many zinc finger domains, some which are involved in protein interaction, others involved in DNA binding. And the critical region for DNA binding is at the C terminus, where there's a cluster of 3 zinc fingers called 4, 5, and 6 here, which bind a novel DNA recognition sequence, TGACC-A, which was not previously in the transcription factor database. When we crystallized BCL11A on this DNA sequence, we obtained the crystal structure shown here, where the major feature is that zinc finger 5 is the predominant finger recognizing this motif. There's some contribution from finger 4, but no contribution in terms of specificity from finger 6. And this is a sort of zoom in of that, which shows that finger 5 actually makes contacts with the G of the TGACC-A sequence. Now, why is that interesting? Well, it turns out that there are rare, naturally occurring H variants in people called HPFH for hereditary persistence of fetal hemoglobin, where there's a substitution at that G residue, in this case to A. There are also other rare variants. And these were actually described in 1985 in these 2 papers, one of which Francis Collins was the lead author as a postdoctoral fellow. And at the time, it was believed that these mutations might affect the cat box, which is the sequence over here, but in fact, they affect the TGACC sequence, which overlapped that, which was not recognized at that time, 30 some odd years ago. So this indicates that this mutation reduces the binding of the factor to the promoter and therefore allows expression. Now, how does the system work? I won't present really any of the primary data, but in fetal cells, the activator, ubiquitous activator called NFY, binds at the cat box and drives expression of the fetal genes. But in adult cells, when BCL11A comes on, it binds a distal site very close by, TGACC, and recruits a co-repressor complex NERD. But probably the most important aspect is BCL11A evicts NFY, probably for steric reasons, allowing the LCR to contact the beta globin gene downstream. What's remarkable about this is that this switch is engineered over a platform of only about 30 base pairs within a locus of 50,000 base pairs. This is really part of the primary switch. We could show that actually that specific TGA-CC sequence was important using the technique of base editing now, changing those C's to T's, which relieves fetal hemoglobin repression and mimics the HPFH state. So BCL11A has several roles. One we think is direct repression at the promoter, eviction of the activator, and probably physical obstruction, but it also recruits the NERD co-repressor complex to actually close the locus and make it inaccessible to other factors. Now I want to get back to the original GWAS that identified this gene because nothing I've told you so far about its regulation of fetal hemoglobin provides insight into what the GWAS was actually scoring. And this was attacked by another outstanding fellow who's now on our faculty, Dan Bauer, who studied the BCL11A locus itself. And within a large intervening sequence within the gene are 3 DNase hypersensitive sites shown in the yellow, which are not present in brain or B cells, which also express BCL11A. And when that's expanded below, one can see that the highly correlated SNPs, which correlate with hemoglobin F expression, are sort of interdigitated with these hypersensitive sites. And this region is approximately 13 kilobases in length. We thought this might be a regulatory element and red cell specific element, which was actually confirmed by transient transgenic studies using a LAC-Z reporter as shown here, showing that the fetal liver is positive with the transgene and obviously negative without. And we suggested at this time that this enhancer might be a site for genetic intervention because it would be red cell specific and therefore any genetic manipulation would be restricted in its consequences for erythroid cells. Now, as I mentioned, BCL11A is also expressed in other tissues. It's expressed in B cells, where it's important for their development. It's actually important for maintenance of hematopoietic stem cells. Been reported to be expressed in mammary cells. We don't know how important it is there, but it's expressed in the brain during development, where it's critically important. So the question then is, how specific is this enhancer? I've suggested it's erythroid specific, but how specific? And we went back to the mouse for these studies and deleted the corresponding enhancer within the mouse. These mice, the homozygote enhancer-deleted mice, are perfectly viable. And in gene expression, the only cell type which is affected are erythroid cells, which have approximately 100-fold reduction in expression. The gene is essentially off, but the hematopoietic cells of the enhancer-deleted mice work fine. Now, we had a large region here, 13 kilobases in length, which is not addressable easily for genetic manipulation, at least in thinking about translation to patients. So Matt Canver, an MD PhD student, and Dan Bauer carried out the experiment shown at the top, which was to pepper the entire enhancer with Cas9 and CRISPR-Cas9 and guides to see whether there was a region of the enhancer that was more responsible for its overall function than another. And what we found was quite surprising, was that in the center of the enhancer, at this site called plus 58 kilobases from the promoter, there's a site at the GATA1 binding site, the site for the major erythroid transcription factor, where if one makes a single cut and sews back the DNA with non-homologous end joining, so an imperfect repair, one approximates the enhancer deletion in terms of downregulating BCL11A transcription. approximating, I'd say about 3/4 of the activity of the enhancer is dependent on this single site. So it's not the whole activity, but the stringent cell specificity of the enhancer really confers safety for any genetic manipulation within this region. And that's the critical aspect of translation. So human genetics converges on BCL11A in two ways. So I think this is a rather unique gene. First, common genetic variation converges on the enhancer and regulates the expression of BCL11A. And in the common genetic variation, the high hemoglobin F phenotype is associated with a very mild decrement of BCL11A expression. Rare genetic variation, however, in so-called HPFH syndrome, targets the gene promoter, specifically the TGACC site. So really, BCL11A is central to the genetics of hemoglobin F. So the question then was, how could we leverage any of this for therapy? Could we reduce BCL11A level to reactivate hemoglobin F? Now, this must be accomplished in hematopoietic stem cells as a form of somatic gene therapy to be durable. And the notion was to attack the erythroid enhancer, at least in one form of the approach, that is to use a guide and Cas9 to hit the Achilles heel, the vulnerable site enhancer, which would reduce BCL11A expression and allow gamma globin expression to increase, and beta globin expression would be concomitantly reduced. And this would only be in the erythroid lineage, and therefore would be quite restricted and safe. Question was, how much inhibition of BCL11A is needed for therapy? Do you need to get it down to the knockout level, or something intermediate would be beneficial? Insight to this was provided by another syndrome, a haploinsufficiency syndrome for BCL11A called the Diaz-Lagos syndrome, which is an intellectual disability syndrome in which patients have a deletion of one copy or an inactivation of one copy of BCL11A. These individuals have a global developmental delay, specifically language problems and many others as shown on the right here. But what was important for us is that these patients have persistence of hemoglobin F. And on average, although there's a widespread, it's about 13.5% is the level in the steady state in these individuals, which is well within the classical HPFH genetic phenotype. So this shows that the hemoglobin F level is exquisitely sensitive to dosage of BCL11A. So certainly 50% would be beneficial to patients with either thalassemia or sickle cell disease. But if we could get it slightly lower, that would be even better. So the therapeutic approaches that have been employed to attack BCL11A could take various forms. One would be the knockout of the gene. One would be a knockdown with shRNA, like the original experiment in culture. One could edit the enhancer, as I suggested, or even mutate the binding site within the promoter that binds BCL11A. The straight knockout is toxic for hematopoietic stem cells and B cells, so it has not been translated. The shRNA approach has been translated at Boston Children's Hospital by my colleague David Williams in a gene therapy experiment where the shRNA expression is restricted to erythroid cells with a globin lentiviral vector. Another approach is to use editing of the enhancer, and that's ongoing. And there are preclinical studies in progress in a number of labs to employ direct attack at the binding site. Genetic therapy of blood disorders involves taking hematopoietic stem and progenitor cells from an individual, modifying them ex vivo in the laboratory, either with a lentivirus or gene editing, reintroducing these products into a patient who's been preconditioned, so their endogenous bone marrow is destroyed, and then infusing these back. And there are many, many clinical parameters that are critical here, including the fraction of cells that are modified, how you modify an allele, the effect of that modification, off-target effects, cell dose, preconditioning. These are all critical and have to be done with precision in order to carry out the clinical translation. Now, the translation has occurred with 2 trials. I'm just a pleasant and happy observer. I'm not involved in either one. And these were reported in the New England Journal just at the turn of last year. One was a trial of gene editing, the first in human gene editing trial for genetic disease by Vertex and CRISPR Therapeutics. And the other was at Boston Children's Hospital with David Williams and clinician Erica Ezrick using shRNA to drive down BCL11A expression. The children's trial is just summarized here briefly with the vector shown at the top. But here's the course of the first patient after therapy, showing a rise in what are called F cells. These are cells that contain substantial amounts of hemoglobin F. And the total F level in this patient was 25% when well out after therapy. This blood smear was normal 6 months later. And this patient is now nearly 3 years out from therapy and has been well without sickle crises and has had a durable response. What's remarkable about the children's trial is the subsequent patients all have a very similar clinical outcome in that they have about 70 to 75% F cells. Each F cell has about 40% fetal hemoglobin. And that's the critical number because that's well above the threshold to block sickling. And these patients have all done quite well. In the CRISPR Vertex Therapeutics trial, they've treated patients with both sickle cell disease and patients with beta thalassemia with very similar, if not even more favorable results in terms of the number of F cells and the percentage of F. And at least by reports, the patients have had a benign course of following therapy. This summarizes what we know so far about the trials that have been executed. I think from our standpoint, they validate the preclinical work on BCL11A as a repressor of fetal hemoglobin expression. In both of these studies, the hemoglobin F level is about 40% in the F cells, which is well in excess of needed to prevent sickling. And there have been disease-altering outcomes, at least by reports thus far. There have been no sickle crises, and the beta thalassemia patients have been transfusion independent. And to date, there are about 28 patients that are in the sort of public domain. No toxicity or adverse effects have been described. There are many more patients, we believe, who have actually gone into these therapies. There are many targets for genetic therapy of the globin disorders, the beta chain. The one that everybody points to first might be correction of the beta globin gene, the point mutation itself by homologous recombination. That's each so-called homology-directed repair. That has problems with the efficiency, although it probably will go into the clinic soon. We'll see how that turns out. There are several BCL11A-directed therapies, either lentivirus, gene editing of the enhancer, the promoter, or even base editing of the promoter. And then very recently from David Liu's lab is the possibility of base editing to change the sickle mutation to another hemoglobin, Makasar, which is a non-sickling hemoglobin. So we believe that the genetic therapies are now validated, at least the approach, and we'll have to see over time how the therapies, whether they're durable and have no side effects of major consequence. But how can they be improved? Because the therapy is invasive and intensive, and involves really a large medical infrastructure. There are two ways that this is being thought of. One is to try to develop non-ablative preconditioning, perhaps with repeated administrations of modified cells, or perhaps only one administration. That, I think, is still a ways off. Further off is the notion of in vivo genetic therapy with delivery of either a lentiviral vector or some editing-based construct to allow this all to be done in the patient rather than ex vivo. That would be a major advance, but there are a number of issues that I think are challenges to that. So over the last more than a decade, then, we've taken a route from genome-wide association studies through to an understanding of the mechanism of hemoglobin switching with B11A as a major factor. And then this work has fortunately been taken up and translated in two trials, one at Boston Children's Hospital, as I described, with lentiviral shRNA, and the other gene editing of the enhancer, and specifically this vulnerable Achilles heel within the enhancer, and that's the work of Vertex and CRISPR Therapeutics. I just want to remind in closing that the burden of the hemoglobin disorders is really global, and it's still unlikely, at least in the near term, that genetic intervention will reduce the burden of disease. So we still have a ways to go. And I think, in my view, this involves the development of small molecules that inhibit the critical components of the switch now that we understand aspects of the switch. And this could be applied in a population-based way, although it's very challenging from a chemical standpoint. In closing, I just want to also highlight really some of the critical components of the story from a genetic standpoint. That first, obviously, GWAS provided identification of B11A. The ability to do conditional knockouts allowed us to show that modification of this single gene in a relevant engineered mouse model is sufficient. Understanding transcription enhancer biology allowed us to look at the cell specificity of control in this vulnerable site. And then the advent of gene editing really allowed discovery of this Achilles heel enhancer. Really, mechanism of action of B11A at the promoter has been dissected really at the nucleotide level. And now we have even clinical application. So the take-home messages I have are really that the mechanism of the fetal to adult hemoglobin switch is coming into clearer focus, and B11A is a critical and direct repressor of gamma globin transcription. The B11A targeted clinical trials validate reactivation of hemoglobin F as both effective and tractable for therapy with disease-altering outcomes. And a challenge there is expanding access and ease of administration of these therapies to the many patients who might benefit. And I think a complementary approach is developing on-target small molecules, which can address the global impact. This is with really elucidation of the mechanism of hemoglobin switching provides the rationale now for targeted drug therapy to mimic with small molecules what we can achieve with genetics. There are many people who have contributed to the work for in the initial phase of B11A elucidation. The individuals here were the main contributors. I've mentioned a couple of them throughout the talk. This is the laboratory pre-pandemic. And I just want to also give thanks to my family, wife Roz, and daughter Jane and her family shown on the right. And I'll stop at this point and be glad to take any questions in the remaining time. Thank you.
Thank you to our lecturer laureate. As a reminder to the audience, you may submit questions via the question and answer function in Zoom. Let me now introduce our discussion moderators, who are both advisors to the Genetics Prize. Aravinda Chakravarty is the Muriel G. and George W. Singer Professor of Neuroscience and Physiology and the Director, Center for Human Genetics and Genomics at the NYU Grossman School of Medicine. Eric Olson is professor and chair of molecular biology at UT Southwestern.
Thank you. First, thank you for that very exciting lecture, Stuart. And the story becomes more interesting every year that passes. As we are waiting for questions from the audience, and Eric and I are going to take turns reading them, I just wanted to start with the general one, reminded by what Arno Matalski said. So there is a quip that anything you want to learn about human genetics, you can learn from the globin genes. So of the many lessons, some of course are specific to the globin system. Which do you think are the most generalizable lessons for all human genes and their diseases?
Boy, that's a challenge. That's like summarizing all of human genetics. I think from the early work in thalassemia, it was obviously the sort of diversity of mutations which can have outcome in a similar phenotype, which I think is well established. I think the notion that certainly haven't talked about today, the one that Hay Kazazian with the notion of haplotypes and association of polymorphisms with specific mutations, which allow dissection of the genes. I think the enhancer studies of the locus control region really predated many of the current studies of super enhancers and the way they function. I would say that the GWAS showing that the major effect is within an enhancer fits with the well-established role of regulatory elements as really genetic modifiers, I think is quite clear. And I guess probably overall, it's the principle that one can take, really basic findings and hopefully translate them. And the blood system has been a great system for translating things from the laboratory to patients.
Thank you.
Okay, I guess I'll ask the next question. Greetings, Stuart from Dallas. I have followed your work closely for decades, as you know, and we've spoken back to back many times around the world on the stage, and I'm so happy to be here to observe your award today. So the first question that's come in is, how can or will these new therapies scale to the vast number of affected individuals in resource-strapped countries? And as you pointed out, many of the patients are in Africa where they can't probably afford these types of therapies.
Yeah. So, I think this is a long discussion. I think there will be improvements in the ease with which one can do conditioning, for example, that might allow patients not to necessarily be in the hospital to be conditioned for, infusion of modified cells. I think the possibility is also building centers where you could scale bone marrow transplant. And this has been done in some parts of the world, scale it so that it could be done at a lower cost and more efficiently than we do in this country. But I do think that to get the burden of disease down, it's a hard road for genetic therapies unless one can do them entirely in vivo, like a COVID shot or a flu shot, which I think for some disorders, the liver, for example, in vivo therapy, I think is being developed, eye therapies. I think for the blood disorders, I think in the hemoglobin disorders, the challenge is modifying enough cells, enough stem cells in this case, to have a clinical effect. And we can argue about what that, or guess what that level is, but it's an appreciable number of cells. And particularly the stem cells are in the bone marrow, they're quiescent cells, they're not cycling. I mean, they have all the attributes of cells that are difficult to reach. And that's why I still think that there's a huge opportunity in terms of small molecule development. And it couldn't be done before we understood the nuts and bolts. And I think now we understand the nuts and bolts. It's a difficult problem, but I don't think can't be solved.
I agree. Thank you. Aravinda, next question.
The next question asks, when BCL11A is modified experimentally, is erythropoiesis stimulated in the liver and spleen?
No, I'm not quite sure exactly what the thought process is here, but the way the modification is done, at least at the enhancer, let's say, the consequences are entirely erythroid specific, so that any other aspect of the hematopoietic system, whether it's B cells or myeloid cells or stem cells, as best we can tell, are normal.
Okay, we have a question from a prior recipient of the Gruber Prize, Dr. Steve Elledge. His question is, what is the status of understanding the HU induction of fetal hemoglobin? Is there a pathway to reactivate that endogenous source that would not require editing? And how does HU work in this respect? There's kind of three questions embedded in one there.
Well, I think the answer is pretty short. I think Steve got the Achilles heel of the hydroxyurea story in that we don't really understand how it works. The best proposal is that it affects something called what we vaguely call stress. If you stress erythroid progenitors, it's been known for many years, they'll make more hemoglobin F. That's the case, for example, after normal bone marrow transplant, when the cells are stressed, repopulating, they'll make more F. And hydroxyurea somehow takes advantage of that. Obviously, it may affect the replication kinetics of the precursor cells or progenitors, but we have no suggestion that it actually acts directly in the pathway of any of the repressors or anything I've been talking about. The other irony of the field is that hydroxyurea is relatively inactive if you add it to the cell systems that are currently used. So although it was first used, and actually Dr. David Nathan, who's probably on this, was really the instigator of the use of hydroxyurea, suggesting it would be less toxic than 5-azacitidine, which was, the notion was a demethylating agent. That was really first employed in primates and then in people, but the preclinical work on it was meager. So I think today, if we started with hydroxyurea, it might not make it very far down the path.
Thanks. The next question is, where are we with small molecule screens? And the question is whether you've done some and whether, given the structure of BCL11A, there are any specific leads.
Yeah, well, that's, I should say I'm sort of turning, although I don't want to say this to the Gruber Committee or the Gruber Foundation, we're sort of turning our attention more to chemistry and structure in my group than to genetics right now. And this is something that I've been working on with the group for a number of years. It's a very tough problem. It's called drugging the undruggable in the sort of chemistry world. But I just say that I think there are opportunities and I think it can be done. I'm rather pessimistic about sort of blind screens, that is just sort of taking collections of compounds and putting them on cells or molecules to try to get a home run. That's like winning the mega bucks or whatever, But I think a focused approach eventually will be possible. If not with BCL11A, with maybe one of the other components.
Thanks.
The next question is from Ross Hardison. The question is, you explained the cell type specificity of BCL11A, but what about gene specificity? Is the expression of other genes in red cells impacted by reduction of BCL11A?
Yeah, what's remarkable about BCL11A, I think we knew from the initial knockout studies, was that the red cell development is pretty darn good, which is not what I've seen in other transcription factors that we've worked on over the years. I think Eric would probably say the same. Most times you knock out a transcription factor, the cells turn over and die or do something else. We've actually recently looked at that, a work that we're currently sort of putting together for a paper. We've used conditional protein degradation to look at this, where you can acutely remove the protein and see what happens to transcription at genes in real time, as opposed to waiting at the end of a genetic experiment. And the remarkable thing is that, if we're generous, there'd be 50 targets of this repressor over the entire genome. Probably it's more like 5 or 10. And so somehow, dumb luck, I call it, this regulates very, very few other genes in the cells.
Thanks. The next question is from Maynard Olson, who also is a prior recipient of the Gruber Genetics Prize. And Maynard asks, does the population distribution of the variants give the GWAS signal any suggestions of evolutionary explanations as to why common variants with such large functional effects exist?
Yeah, one thing I did not emphasize was that these common variants are distributed through all populations, at least that have been studied. So it's not restricted to particular disease populations or geographic regions. There is variation in the sort of frequency of the variants in different parts of the world, but all populations have these 3 loci as hits. I don't think we know anything about sort of the evolution of it. At least I don't.
No. Thank you. Eric?
Steve Elledge has another question here, and that is, what is the current status of the search for small molecules to promote refolding of hemoglobin, as has been done so successfully for cystic fibrosis? Has there been an effort to use more modern library methods to achieve this goal?
That's another good question. There is a company in California, in San Francisco, called Global Blood that has an recently approved drug called Veloxetor, which really stabilizes sickle hemoglobin in sort of the oxygenated state. And although this is approved, it was approved on the basis of a phase 3 trial, which increased hemoglobin level 1 gram. It was not approved on the basis of a therapeutic benefit to patients. I would say it's a modest advance. I don't think it's solved the problem, but I think it is it's still a viable idea, is to try to use a small molecule and structure to fix sickle hemoglobin in a proper conformation. So it's been done part of the way. This is actually sort of the original idea of Tony Cerami with urea compounds and cyanate in the old days.
So the next question, Stu, is a general one, but perhaps you could tell us a little bit about what's been the status or effectiveness of premarital carrier screening and genetic counseling for prevention of, say, sickle cell and other hemoglobinopathies.
I think the story for thalassemia has been very successful, and particularly in places like Sardinia and other parts of the world where there's been huge carrier identification and prenatal diagnosis and counseling. And that's been advanced even by the church in Italy initially, but it's been a dramatic decrease in new births. I'd say in sickle cell, there are a number of issues which have really confounded that, and I don't think there's been as much advance.
Okay, the next question is from Claudia Carvalho, and the question relates to the potential role of BCL11A in the brain. And the question is, during the tests being performed for the genetic therapy of sickle cell disease, is there any possibility of the function of BCL11A also being affected in the brain? Are long-term side effects being considered in these therapies?
Yeah, there's definitely a role for BCL11A in the brain during development, less known in the adult. The beauty of the ex vivo stem cell therapy for genetic disease is that the cells are hematopoietic cells and stay in the blood, and in this case, the editing are only editing an enhancer that's active in the hematopoietic cells in the erythroid lineage. So there is no effect in the brain. I think this applies more if one thinks about small molecules, where one might want to keep them out of the brain, but many of the chemists I talk to say it's easier to keep things out of the brain than put them in the brain. So that'll have to be addressed at another time.
Thanks. So but just to clarify, Steve Allage was asking about refolders for hemoglobin, but you addressed that. But there's another question from an attendee. Fascinating question. It said, if you were early in your career and looking at this field of research, what major unknowns or grand challenges would you choose to tackle? And what do you think the next generation of researchers should do to take it forward?
If I were doing it
basically,
now,
what are the hot questions that you think you would focus on that are really interesting?
Yeah, well, for myself, I think the one is in the application is really making genetic therapies more translatable in terms of making them easier, less expensive, less cumbersome. I mean, I think that's a major challenge, and that's going to take many, many decades. And I think the other one is actually on target small molecules. I think, that's still, I think, a big one, because I think there the spin-off could be in other areas as well. I think the more we learn about how to do that, the more it'll have impact in other fields.
Thank you. I'm going to turn it over to Eric, who can say thank you again. This has been a fascinating lecture and great participation by the audience. Eric?
Yes, congratulations once again, Stuart, on this well-deserved recognition of a lifetime of achievements in this field. We are deeply appreciative of your work. I'll turn it back to Sarah.
Thanks to all three of you for the discussion. And if you'd like to stay on screen, we welcome you to do so. It is now my pleasure to bring on our closing speakers, a few special guests to end these proceedings on a more personal note. First, let me welcome David Nathan, President Emeritus, Dana-Farber Cancer Center, Physician and Chief Emeritus, Boston Children's Hospital, and Robert A. Stranahan, Distinguished Professor of Pediatrics, Harvard Medical School, to say just a few words about our recipient, Stuart Orkin. Professor Nathan.
Well, thank you so much. I hope you hear me. This is just a wonderful day for Stuart and Roz and the Orkin family. It's a great occasion for the Harvard Medical School and the Boston Children's Hospital, the Dana-Farber Cancer Institute, and the Howard Hughes Medical Institute. I only wish that Phil Leader, who played such an important role in Stuart's career development, could be with us today to celebrate such a marvelous occasion. It's particularly satisfying that the Gruber Foundation Award is based at Yale, where medical genetics has been so greatly advanced. Finally, it's a day that gives me enormous personal pleasure. I've been in the business of training physician scientists since 1959, when I opened my lab at the then Peter Bent Brigham Hospital and later moved it to Boston Children's because inherited diseases of the blood so fascinated me. I'm always on the lookout for very bright, scientifically adept interns and residents. And one day in 1972, I met Stuart Orkin, an intern then at Boston Children's, who was about to go to NIH to work with Phil Leader. I was so impressed with him that I asked the incomparable Charles A. Janeway, a Yalie, our pediatrician in chief, whether he would recommend him for appointment to my hematology division. Janeway responded with one of his famous two-liners. Stuart Orkin, he wrote, is a small man with a very large mind. Take him. When Stuart returned from NIH and completed a year of residency, I gave him his own lab immediately. I've never done that for any fellow before, nor have I done it since. I shortened his clinical training to an absolute minimum. Above all, I left him alone, other than to sort of put my head in the laboratory and ask him occasionally if he ever needed anything. I did give him blood samples from patients I saw, but that was with the hope that he would do something intelligent with them. When I asked him if he needed anything, he always said no. He was and remains the most brilliant physician scientist I've ever seen. And on top of that, he's married to Roz Orkin, who's his intellectual equal. No one in his lab is his technical equal. Roz now counsels our young trainees. She too is a blessing. It's obvious that I'm terribly proud of Stuart, that he became the inaugural David G. Nathan Professor of Pediatrics at Harvard fills me with absolute joy. Now I'm reveling in the Gruber Foundation Prize. And I want to thank the trustees of the Gruber Foundation, the Yale School of Medicine, and surely Stuart and Roz for this marvelous day. Thank you for asking me to make these comments.
And thank you, Dr. Nathan, for this lovely tribute. And Professor Orkin, if Roz Orkin is in the room, she's welcome to come on camera with you. That would have been fine.
I know she's getting a flu shot.
My goodness. Probably a prudent choice. Well, let me just call for our final words on our co-founder and president emeritus, Patricia Gruber. Pat?
I think I'm unmuted now, yes?
There we are, yes.
Okay, great. So on this happy day, let me add my personal congratulations to those that have already been heaped on your worthy self. Your peers have unanimously and enthusiastically selected you with much rigor from an impressive field. Your colleagues have sincerely sung your scientific praises, both of your work and your person. We congratulate you again. It's really a deep satisfaction to me to see the vision that was shared by my husband and myself continue to flourish and evolve in honoring these insights in genetics. We thank you today for giving us science worthy of the highest recognition. This concludes my remarks. Thank you.
Thank you. She's
Thank you,
arrived with a flu shot.
Yay!
just in time.
It's perfect.
Thank you very much. Thank you all. Thank you, Mrs. Gruber. Congratulations again to Professor Orkin. And thank you all for attending. This concludes the 2021 Gruber Genetics Prize.
Thank you.
Thank you. Thank you.